Hello students, in last two lectures we have discussed about the auditory system and the electrical activity occurring in the auditory system. So today in continuation to the two lectures we will discuss more about auditory evoked responses. So what are basically auditory evoked responses or evoked potentials? Evoked potentials are the electrical signals that are generated by the nervous system in response to a stimulus and are event related. That is, they are evoked by the onset of stimulus. With evoked potentials, it could be a number of stimuli. It can be visual, it can be auditory, it can be some sensory stimuli. Here we will mainly concentrate towards auditory evoked responses. The selection of presentation stimuli is dependent on what type of evoked potential you are performing. So evoked potentials are mainly useful in diagnosing a variety of neurological disorders. So let's now move towards auditory evoked potentials, that is mainly when the stimulus is a auditory kind of response. They are also known as auditory brainstem response or brainstem auditory evoked response, because as we know that auditory system is mainly what part of brain is involved, it is brainstem. So auditory evoked potentials is a diagnostic tool to determine the integrity of the auditory system and make inferences about hearing, because it deals with hearing, so basically it makes inferences about hearing. Remember, performing auditory evoked potentials is not a hearing test, but it is a tool to predict hearing. Also, with auditory evoked potentials, or in short we can call them as AEPs, are an acoustic stimulus generates a response measured using skin electrodes on the surface of the skin. The stimulus that is being generated is minimal. Thus, we have to use gain and amplify to measure the response. This all we have also studied in last lecture, that because the gain is less, therefore we need more and more number of observations in this, and we have to amplify the signal, maybe 1,000 times of the input. It also objectively tests the integrity of the hearing system from the level of the cochlea to the brainstem. Brainstem auditory evoked responses or auditory evoked responses are electric field potentials generated with repetitive auditory stimulation of the auditory pathways, and they are reproducible electrical potentials that are elicited and recorded from scalp electrodes. It is considered as an exogenous response. Why it is considered like that? Because it is dependent on the external factors. It reflects neuronal activity in the auditory nerve, cochlear nucleus, superior olive, and inferior follicles of the brain stem. The measured recording is a series of 6 to 7 vertex positive waves, of which 1 through 5 are evaluated. These waves are labeled as Roman numerals in Jude and Williston convention, that is the standard convention, occur in the first 10 milliseconds after the onset of an auditory stimulus with an amplitude of 1 microvolt. As I mentioned already, due to their small amplitude, as we have seen around 1 microvolt, we need 500 or more repetitions of the auditory stimulus that are required to average out the random background electrical activity. The auditory structures that generate the auditory brainstem response are as you can see in the diagram. So we have wave 1 through wave 3. They are generated by the auditory branch of cranial nerve 8, or we can say 8 nerve and the lower. Wave 4 and 5, they are generated by the upper brain stem. Now we will more see in the depth location where actually those locations are from which these type of waves are emerging. So wave 1 originates from the dendrites of the auditory nerve fibers. Wave 2, it will originate from the cochlear nucleus. Wave 3, it shows the activity in the superior olivary complex. And wave 4 and 5, they are associated with the lateral lemniscus. Now, how we will measure, how we will record these signals. So where we will place the electrodes. We have seen that we have to place the electrodes like we are doing in EEG. But let's find out the basic locations where we are placing the electrodes to get this signal. As you can see in this diagram, we have CZ, A2, A1. So one electrode we will place on the scalp at the vertex. That is basically CZ position, that is position on the central, or we can say midline position of the 1020 international system of EEG electrode placement. That already we have seen the 1020 international system of EEG recording. So in that we know the position CZ. So at that point we are keeping one electrode, and then second electrode we are keeping over the left and right earlobes. You can see A1 and A2. They are again the positions of the 1020 system, 1020 international system of EEG electrode placement. Or they can also be placed on the left and right mastoid process that are signified as M1 and M2. Now what are the main recording parameters? So we have to take impedance that should be 5 kilo ohm or less, equal between the electrodes. The filter settings should be, it should be 30 to 15 hundredths of bandwidth. Time window should be minimum 10 milliseconds. Sampling rate we are choosing to be high sampling rate of 20 kilohertz. And the intensity usually start at 70 decibel for normal hearing level. Stimulus is, it can be a click, which is approximately 100 microsecond long. It can be a chip type of signal, or it can be a tone burst. And the transducer type, that can be inserting, means that can be an invasive kind of electrode, it can be a bone vibrator, it can be a sound field or headphones. And as we mentioned, we need amplification, that is approximately 100 to 15K of the amplification which is required. And what are the minimum number of we can say observations we need? That is denoted by small n, and that can be 1000 minimum observations, or it is recommended to have 1500 observations. Now let's discuss how we interpret the results depending upon the waveforms obtained. So amplitude will determine the number of neurons that fired. Then latency will determine the speed of transmission. Inter-peak latency will determine the time between the peaks. Interaural latency will define the difference in wave 5 latency between the ears. The ABR represents initiated activity beginning at the base of the cochlea and then moving towards the apex over a 4 millisecond period of time. The peaks largely reflect activity from the most basal regions on the cochlea because the disturbance hits the basal end first, and when by the time it goes to the apex where a significant amount of phase cancellation occurs. Now we'll see this wave 1. It should be observed but will only be present ipsilaterally. Then you can see wave 3 and 5. They should be detectable in all the healthy individuals. Wave 3, it is often has decreased amplitude, as you can see on the side contralateral to that being stimulated. Then wave 6 and 7, they appear variably after wave 5. Most of the cases they are not there, in some cases they are there. And wave 2nd is often absent. And if you see wave 4, it is frequently buried and therefore indistinguishable from wave 5. So this is all in this. We have seen how we can interpret the results, how we can capture the results, means where we can place the electrodes. It is mainly using again EG as EG 1020 system for pointing the electrodes or the positions of the electrodes where they have to be placed, and then we have seen what the different waveforms signifies. This was all in this lecture. Thank you so much. These are the references we have used. So thank you so much.